Hi everyone, my name is Jason Slingsby. I'm CEO at Tozaro and really pleased to give you an introduction to what we're doing with Smart Polymers to improve viral vector purification. So for those of you who don't know us, we're 30 people based in the UK just west of Cambridge and supported by our investors. So the challenge as we see it in the cell and gene space is a lot of programs have viral vector manufacturing costs in the multiple millions per batch, and there are few downstream purification products designed specifically to purify viral vectors. Historically, a lot of focus was on viral clearance for chromatography. So downstream purification remains poor with around 30–40% recovery rates, which increases cost per dose, means you're throwing away over half of the material that you've made diligently in your upstream. There have been affinity ligands in the field for a number of years, primarily antibodies and peptide-based approaches, but they don't seem to have addressed the majority of customer needs. So we're looking to innovate in this sector to create more value for all and increase patient access. So our solution to this is called the Smart Polymer platform, and this focuses on designing affinity ligands in silico for optimal characteristics to be used explicitly in chromatography. We can use it for rapid analytics, but I won't be focusing on that data in the presentation today. So having come from the CDMO sector myself at Oxford Biomedica and having sat in a lot of client meetings over the years, we feel that what the market really appreciates is firstly a rapid design cycle, the ability to come up with affinity reagents quickly in a matter of weeks and months and not many months or even years. People want a gentle binding and gentle elution mode. Viral vectors are delicate, and ligand stability is really important, so ability to use range of conditions and temperatures. Cost competitive is super important. Chromatography solution providers need to think about manufacturing scalability and potentially reusability as is used in the biopharm protein A chromatography field. So what is a smart polymer? On the left, you see the world of proteins and peptides. As we know, focused on 20 amino acids, put in specific orders with the ability to incorporate one or two additional non-canonical amino acids in the synbio sense. Smart polymers are made out of chemical monomers that can be efficiently polymerized into linear chains, but there's a vast chemical variety of side chains that we have identified and put in our proprietary curated database. So things that amino acid side groups just don't have, such as switterionic and halogenated or even biotinylated. So this is a really big idea company, the ability to generate non-protein binders de novo against specific protein epitopes. So how we do it is this. We start identifying protein epitopes that we want to target. We do in silico docking of these 500 monomers to identify strong specific interactions. And we have incorporated a machine learning loop using real-world lab data to increase the predictiveness of the platform. We'll synthesize a couple of dozen, maybe up to 50 polymer designs based on the output of the in silico design, screen them for optimal characteristics, and come up with some hits. So the vast majority of the design process is done in silico, but clearly everything's focused on picking lead designs and lead hits. So the first program we're working on is affinity chromatography for lentiviral vectors. VSVG is the default envelope protein of choice for where the ex vivo CAR-T market is currently. So we identified 7 epitopes, 91 monomers were identified as potentially having specific interactions, and we only synthesized 18 candidates. And looking at optimal binding conditions, we achieved high capacity binding with 4 linear polymers, and of those 3 were optimal eluters, as in eluting more than 65% of bound product. So we're really pleased to share this proof of concept data. So this is a chromatography trace showing our linear polymers immobilized on a membrane-based chromatography support. And this was raw, unprocessed, crude harvest material, no benzonase treatment. So very much a kind of tough challenge. We shifted the pH down from 7.6 to 6, so very mild shift. And the first peak you can see with all of the character lentis is the peak that contains the active lenti. So we loaded with a high flow rate, which is really important, had good membrane capacity, and 50% recovery by particles by P24. And vast majority of that was functional lenti, so really good TU to P24 ratio, good product quality. What was really exciting is the very clear reduction in host cell proteins of 99.8%, and the toughest challenge for a lot of viral vector downstreams is removal of DNA, which is 99.9% removed. So super excited by that. Clearly, standard methodologies currently use anion exchange chromatography. So these are all preliminary numbers. We've not finished final product configuration, but we see clear differentiation where we can seek to have good capacity, more gentle elution conditions, really good recoveries, but potentially most differentiating very strong removal of DNA and unwanted host cell proteins. And back. So an even harder design challenge would be a smart polymer that can bind to all AAV serotypes. Of which there's approximately 12, 13 major families. So this was the tougher in silico design challenge, but we believe because the binding mode is sort of a linear polymer that's very flexible without secondary or tertiary structure, we can go places that antibodies and antibody fragments would struggle to sterically. So we identified 5 shared epitopes across all major natural AAV serotypes, similar number of monomers identified, and 30 linear polymer candidates. And then we identified 6 pan-AAV binders. So I'll show you the data, 2 of which show really good elution characteristics. So once again, these are not final numbers, but on the left you can see a reduction in AAV quantities in the flow-through, which means binding. So that was tested across AAV 1, 2, 5, and 9, where most of the phylogenetic tree is covered, and very equal levels of elution as well. So this is one candidate across a number of lead polymers, but we feel that there's a significant opportunity to be second and better in the AAV affinity purification field. So we're pleased that we're seeing really good commercial traction for adoption of this novel affinity ligand platform within the industry. Our first announced collaboration is with Sartorius, actually in the rapid analytics field. So we've immobilized even higher affinity polymers of this family on the Sartorius Octet BLI instrument, which you can see near the meeting rooms. And we're able to do a 2-minute titre measurement for both VSVG lenti and AAV which opens up the potential for inline decision-making to be incorporated into GMP manufacturing protocols. So we're interested to follow up on that in the coming months as well. When it comes to chromatography partnerships, we've got additional undisclosed collaborations to access some of the industry's leading chromatography supports. As any DSP gurus among you will know, there's a wide variety of technologies. We've shown this proof of concept on membrane-based chromatography, but we're also accessing leading monoliths and bead-based matrices. And we're relatively agnostic. We want to have the best product performance for our smart polymers and the vector category. Finally, this is a platform technology company. While with 30 people, focus in product development is super important, we do have the ability to take challenging affinity purification challenges much more broadly within biopharm and rapidly generate candidate non-protein binders. And yeah, we're the only people optimizing protein polymer interactions. The rest of the sector is focused on protein design, de novo protein binding. So I think this is a real differentiator for us in the market as well. So in summary, I can take my time. I've got 3 minutes. Tozaro is developing the Smart Polymer platform for rapid affinity ligand design. While we love generating data in the lab, we really like the fact that the majority of the discovery process is done in silico, leveraging machine learning. And it's chemistry, and chromatography technology developers are also chemists. So this is a animal origin-free, cell line-free chemical optimization opportunity. Really importantly, this is polymer chemistry, so it's cost-effective and scalable in manufacturing, and we do see some really interesting purification as well as capture and release. We've shown proof of concept for the 2 leading off-the-shelf products that are required in the cell and gene sector, hence us being here at Meeting on the Mesa. But we do see application and indeed inquiries on other complex challenges. There's still a lot of purification challenges in the field of DNA, RNA therapeutics, complex biologics where protein A fails as an affinity ligand, and potentially even in vaccines. So the kind of news we hope to be sharing with you over the coming months includes raising further equity from current and new investors. So yeah, we're currently backed by 4 UK VCs. Very importantly, signing corporate partnerships to access leading chromatography formats and indeed the leverage from global sales and marketing capabilities. We're also expecting to have a 2026 launch of a research use only or RUO kit to let innovators and process development groups utilize the simplicity of these affinity ligands and to help seed the technology at an innovation-friendly community. And we are looking finally to have further technology and product proof of concepts to show that this is a broadly applicable, scalable, cost-effective affinity ligand platform, not only relevant for cell and gene, but more broadly in the biopharm industry. And with that, that's where we are. We're west of Cambridge in the UK, and yeah, hope to see you again this time next year. Thank you very much.
Thank you, Tozaro.